The International Space Station is used by all of the partner space agencies for scientific research that can't be done in an environment where gravity is dominant. In the case of some medical experiments, the weightlessness allows cells to behave in ways that help researchers learn about the nature of cancer cells, which is information that can help in the fight against the disease. One recent example of that is an experiment known as cell box thyroid cancer. And to tell us about it, we're joined this morning by the principal investigator, Dr. Daniela Grimm of the Department of Biomedicine Pharmacology at Aarhus University in Denmark, and her colleague, Dr. Markus Wieland of the University of Magdeburg. Dr. Grimm, you had a previous study on the station in which you found that tumor cells behave a little less aggressively in space. Do we know yet why that is?
We use a polydifferentiated thyroid cancer cell line, which is a very aggressive tumor. This tumor cell line comes from a lymph node metastasis from a 42-year-old male. And this experiment, which we had on a space flight before Cellbox, showed that these cancer cells produces a lot of genes and proteins which were regulated in a way that it seems that they promote to shift to a less malignant form. We don't know what is the reason for it, because it was first time done, and it was an empiric observation first. But we have found that these cells grow three-dimensionally. That means like a ball, and these are round aggregates. We call them spheroids, which are very similar to a metastasis in the blood or a microtumor. They are nearer to the in vivo situation. And the primary focus of Cellbox was that we wanted to repeat the experiments and further elucidate the underlying mechanisms.
So that was what you're hoping to build on with this current experiment.
Yeah, this was the main purpose of the Cellbox experiment, was to reproduce the results which we had obtained from the former space experiment. And another important point is that observed effects, we have to exclude that they are not artifact, and we have to reproduce them. One important point was that we wanted to focus on the proteins which may be involved in 3D growth formation, which are involved in spheroid formation. And during the Cellbox mission, we have found interesting proteins which might play a role in the inhibition of this 3D growth. And such proteins are related to a matrix around the cells. It's called extracellular matrix, where a lot of signaling between the cells takes place. And currently we are working on secreted proteins and have some interested data which we will publish in the near future.
Your experiment flew to the station on a Dragon cargo ship about 18 months ago. Tell us about what samples it was that you used, that you sent to space, and what was done with them while they were there on the space station.
This was a cell line, a thyroid cancer cell line called FTC 133, which we studied in detail near the last 10 years. And it's well known in the scientific research area. The cells were seeded in the Cellbox containers and incubated for 24 hours in 37 degrees and with 5% CO2 overnight on ground in the laboratory. And the next day, the hardware was checked by Airbus and the engineers. Then was the handover to NanoRex and NASA. NanoRex and NASA people transported the containers to the launch site and integrated the samples in the Dragon spacecraft. After the arrival on the ISS, the hardware was installed in the NanoRex facility, where the cells were kept at 37 degrees. And after 10 days, the cells were fixed with a special fixative, we call it RNA-later, for later gene analysis. In addition, we collected the supernatants, the medium, which was analyzed by a United States company. It's called Myriad RBM in Austin, in Texas. Yeah, and after when we got everything back from the Dragon, we analyzed the data.
And you've had them back for some time now. Is there anything that you've seen so far that is encouraging to you?
Yes, we have identified proteins. We have published this also in Proteomics this year, which are involved in the negative regulation of spheroid formation. And in addition, we have found a variety of biomolecules of secreted factors by the cells, which were found in exerted elevations in space samples, not on ground samples. And many of these enzymes which were secreted are responsible to degrade these extracellular matrix proteins, which we have found also in the proteomics study. And this is what we do right now. We publish these samples, these data in the near future.
Now, most experiments on the space station involve collaboration from a lot of different entities. Dr. Wieland, tell me about what it took to get this particular experiment together and get it to orbit and get it back.
Yes, this was indeed a collaboration of many people involved, and one of the most important roles were, of course, taken by the German Space Agency DLR, who offered us the opportunity to take part in this mission. And they, of course, were collaborating with NASA and SpaceX. So thank you to all those companies and institutions who offered us the opportunity. Then there was Airbus Defence and Space, who designed and constructed the cell culturing hardwares to our specifications. Space Life Sciences laboratories provided us a laboratory space near KSC where we could prepare for the flight, culture the cells, and of course see the hardware. Then NanoRex offered the infrastructure on board of the ISS, where all the cell box hardware could be housed and incubated at the right temperature. And of course, finally, and last but not least, we would also like to extend our thanks to our universities of Aarhus and Magdeburg, who always supported our research.
Dr. Grimm, are you planning for future experiments to try to follow up on what you've learned so far?
Yes, we have on SpaceX 8 the ESA Spheroids project. Today we had a teleconference and we heard that the prospected launch date would be in January 3, early this next year. On this mission, we will have endothelial cells and we will focus on three-dimensional growth, and we will also see whether some growth factors can induce tube formation. We also will focus on several mechanisms of cell death and of differentiation and cellulation changes of these cells.
Well, Dr. Grimm, Dr. Wieland, good luck with your work. Hope you have success as you continue. Dr. Daniela Grimm is the principal investigator of the cell box thyroid cancer experiment, and Dr. Markus Wieland, a member of her team.